yours, Fabian.
Great. Thanks so much for the kind introduction. It's a great pleasure to be here, and also thank you to the organizers for giving me the opportunity to speak to you today. So as Matteo mentioned, I recently joined the CRIC as a group leader, and I'll tell you first a little bit about what my lab is going to do in the future, and I'll present you one story that I worked on during my postdoc with Peter Sorger at Harvard Medical School. So for me, mathematical models are a pretty powerful way to encode our mental models of how biology works. You turn that into reaction diagrams, can then formulate ordered differential equation models that can be then simulated and compared against experimental data. And for me, this is a pretty powerful way to quantitatively check whether our understanding of the underlying biology is consistent with what we see in experiments. If you want to take this a step further, you can also look at predictions, and in a Feynmanian spirit, I would argue that we do not understand what we cannot predict, and you of course should keep in mind that the reverse doesn't hold, so you can do predictions without understanding anything about the underlying biology, as you see in the case of large language models. Now, the context where I usually apply this is protein signaling, and back when I was a PhD student and attended some of the first ISMB conferences, I had a lot of faith in these pathway or signaling maps, and nowadays I see them a little bit more like these world maps from the 11th century, where you see the UK on the bottom left, you see Europe top right of that, you see Asia on top, and you see Africa on the right, and this is not really consistent of how we would see the world nowadays. In the age of Google Maps, we have a different thing. We put the north of the maps on the north, and overall the shape of most of these continents is different. And for example, you have America just completely missing from the picture here. And I think 900 years from now, we'll probably have a similar picture of how we see biology right now, but hopefully it'll not take us 900 years to get to a better understanding. And one particular aspect of these maps that I want to point to is that they used to put these lines in these maps just to indicate that the cartographer had no idea what was going on in the specific location, and to maybe discourage people from going there, because people were pretty scared of lines back then. And we don't really do that in biology, and that's kind of a pity. And mathematical models often help you to find these gaps in our understanding, because you then ask biologists, how is this actually working? And only then they'll say, oh yeah, I don't actually know. And for me, the way that I think we should go about resolving these gaps in our knowledge is, and I think Eberhard already nicely introduced this morning, is that biology really happens at multiple different scales, and they're particularly interested in structural, molecular, and system scale. And the idea is to use all of these perspectives to come to a consistent picture and connect these different layers with the idea that they're connected as emergent properties of the lower layers. The idea is to use machine learning together with mathematical model in order to make these connections. Now I'll go into, this sounds all a little bit abstract, so I'm going to talk about one specific application that I did together with Luca Girosa, who's a fellow postdoc in the Sorger lab, and this is in the context of adaptive resistance in melanoma. And to just give you a little bit of an introduction, there's one specific biomarker that is typically used in the clinic for melanoma. This is the BRAF V600E mutation. This occurs in about 50% of all patients, and the first-line treatment for this kind of cancer is a combination of the RAF inhibitor vemurafenib and the MEK inhibitor cobimetinib. And they both hit the MAPK signaling pathway, which you see on the right where the mutation occurs in the BRAF gene. Now, the issue with these targeted treatments is nicely illustrated by this case study here, where I show you a patient with metastatic BRAF V600E melanoma. You see all of these small bumps or metastases from the cancer. The patient receives melanoma, the RAF inhibitor vemurafenib, and after 15 weeks, all of the bumps have disappeared, and it looks like the patient is cured. However, just 8 weeks later, you see relapse of the disease, and the patient ultimately dies. And the thinking currently in the field is that this is really caused by a phenomenon where we initially are able to kill the vast majority of all cancer cells, but where we remain with a small subpopulation of cells that can persist during treatment, that gives rise to the clinical phenomenon of minimal residual disease, and then we ultimately see a relapse of the disease from this population of cells. And especially in recent years, what has been identified is this concept that we have first, that this is really a two-stage process, where we first go from a parental cell state to a drug persister cell state in a reversible manner, meaning that if we remove the drug, the cells will actually revert to the parental cell state. And this persister cell state is usually characterized by a slowly cycling phenotype. And then only through multiple rounds of proliferation, these cells are able to acquire new resistance mutations that then confer fully resistant phenotype, where you go back to full proliferation and the disease ultimately relapses. So the purpose of this talk, I'll primarily focus on this initial phase of adaptive resistance, but I'll briefly touch upon the acquired resistance as well. And the experiments that we typically look at is here nicely illustrated. This is wet lab work done by Luca Gerosa, and on the left you see this you see cell cycle reentries, and this is for A375 melanoma cell lines, which were treated with a RAF inhibitor vimurafenib for about a day. What you see is that these cells do re-enter into the cell cycle, which are indicated by these magenta dots, but they do so on a time span of about 3 to 4 days, which is really much longer than cells in drug-naive conditions. And what we then did is we correlated these cell cycle re-entry points with phosphor-ERK pulses that we observed experimentally, and we see a strong correlation between. It's not a one-to-one relationship, but if you pharmacologically inhibit this, and this is data that I'm not showing you here, if you pharmacologically inhibit these pulses, what you see is that these cell cycle re-entry events completely disappear, really suggesting that there's a causal relationship between the two. Now, the interesting aspect is here, not that ERK phosphorylation is linked to proliferation, but that usually with a BRAF V600E mutation, you would expect to see really high phosphor-ERK levels and no pulsatile reactivation. So the fact that we see a decrease in phosphor-ERK levels suggests that the drug is actually doing its job. It's inhibiting the signaling activity from BRAF V600E, but still we see pulsatile reactivation of the same pathway, which is a little bit confusing, suggesting that the drug actually does its jobs and doesn't do it at the same time. Now, the challenging bit about sporadic pulsatile reactivations is that it's really tricky to study this experimentally. So what we came up with is a strategy to study this behavior at bulk level. And this is now shown in this experiment, where we have time after EGF stimulation on the x-axis and phosphor-ERK levels on the y-axis. And if you then, this is now the baseline signaling is after a 24-hour pretreatment with the drug. What you see is if you increase the drug concentration, which is here indicated by different colors, you decrease the phosphor-ERK levels. This is what you expect. But if you then stimulate these cells with EGF, you see a pulsatile reactivation on a time scale of about half an hour to an hour, where the amplitude of these pulses actually increases with increasing drug concentrations. So the question is now, how can we explain this anti-proportional behavior that we saw both in these bulk experiments as well as the single cell experiments on the previous slide? And in order to explain this, we decided to build a mathematical model of the signaling transduction pathway. And this is here recapitulated in this sketch on the left. What you see is that we effectively describe receptor signaling from the EGFR receptor that goes through adapter proteins and second messenger proteins, and when after activation induces dimerization of RAF. RAF dimers can then phosphorate MEK, and MEK can phosphorate ERK, and downstream of ERK we describe multiple transcriptional adaptation processes that regulate the abundance of different proteins and thereby control signal transduction, both at the receptor and the MAPK signaling level. On top of the receptor channel, we also describe an oncogenic channel where MEK is phosphorylated independent of upstream receptor activation simply by mutated BRAF V600E. We encode all of this in a rule-based modeling framework and instantiate this as an ordinary differential equation model with about 7,000 species and about 100 parameters that have to be calibrated in experimental data. This calibration is carried out on training data from around 200 different experimental conditions, which includes data from immunofluorescence, transcriptomics, proteomics, and phosphoproteomics, all in time and dose response to 5 different RAF and MEK inhibitors. And we extensively validate this on really a large set of different conditions, some of which I'll show you over the next couple of slides. And the key why we end up with this really highly complex model is that we have a very detailed description of this oligomerization between RAS, RAF, MEK, and the two different inhibitors that we have in the system. So the idea is to really put all of our biochemical and structural knowledge into this model, then have a little bit more of an unbiased approach to understand what happens to the system in terms of a molecular process. The way that we do this, to cut a very long story short, is then eventually summarized in these rewiring diagrams, where on the left you see kind of the skeleton of the key essential signaling steps that we have in the system, where the receptor-based signaling is in orange and the oncogenic signaling is in blue. What you then see on the right is the instantiation of these rewiring diagrams for different drug concentrations, and this is not just a sketch of the signal transduction, but these are actually computationally derived from model simulations. And what you see is that on the one hand, we have the node opacity, which indicates the signaling activity, so the concentration of the phosphorylated form of EGFR, for example, and then the opacity of the arrows indicate the strength of signal transduction. And this is really an orthogonal measure, as you see on the bottom right, where you have no activity at the phospho-MEK level, but see a strong potential to transduce signals to phospho-ERG, which we believe is also a signaling vulnerability. And we have some evidence that this is actually also then exploited by acquired resistance mechanisms that we see in patients. Now, overall, what this picture should show you is that we see a smooth transition from primary oncogenic signaling in these parental cell states to primary receptor-based signaling in drug persister cells. And what should be clear is that for all of these RAF inhibitor concentrations, you see an activation of the receptor. So it's not about activating the receptor, but relieving the negative feedback. And going a little bit more into detail here, what we effectively identify is two separate mechanisms that I'll explain you a little bit more in detail over the next two slides. This is specifically transcriptional adaptation, so the negative feedbacks that are downstream of phospho-ERG, as well as allosteric interactions. In terms of allostery, what you have to know is that when these RAF proteins form dimers and are targeted by the drug binds to a specific conformational state where the alpha C helix is an out conformation. And due to steric clashes, the second protomer in a RAF dimer can be in the same conformation, meaning that you can target both protomers in a RAF dimer simultaneously, and you effectively end up with partial inhibition of the signaling system, at least in the case of RAF dimers. Now, in the oncogenic channel, the BRAF V600E doesn't require dimerization, meaning that we can potently inhibit RAF signaling in the oncogenic channel with only monomers, as indicated by this blue curve here, and we have this protection from signaling, from inhibition in the receptor channel here indicated in orange. And we only encode the general mechanisms here in terms of allostery using a thermodynamic framework that encodes the relative affinity in terms of Gibbs free energies in a very thermodynamic fashion, and the model then, or the model training then learns really the extent of these different properties from phosphorylation data without any insights into the exact structural properties here. Now, the second ingredient that we need to understand the system is transcriptional adaptation, and I'm here showing you the transcriptional feedbacks or abundances downstream of phospho-ERG. DUSP is in blue, Sprouty is in orange, and EGFR is in green, and darker colors indicate the protein abundances, lighter colors indicate the mRNA abundances. On the left, you see the time course for drug adaptation. So this is the stuff that happens in the first 24 hours. And we see that these drugs adapt their new drug-induced state on a timescale of hours to days. And if you then compare this with EGF simulation, which happens on a timescale of 2 hours, you see there's a little bit of change at the transcriptional level, but this doesn't really propagate to the protein level, indicating that these adaptive processes really create a distinct signaling state that persists throughout these pulsatile reactivations and are not changed throughout this process. What this really means is that this timescale separation between signal transduction and transcriptional adaptation conveys the system with memory and really allows the system or the cells to adopt new cellular states. One specific aspect that you should note here is this green curve, where you see a decrease in EGFR concentration in response to EGF stimulation. And what we believe is happening here is that you see endocytosis of the receptors and ultimately lysosomal degradation of the receptors. And we have additional evidence that this is actually true, and that by investigating EGFR amplifications and then stimulating the system with EGF, which allows us to go to a sustained activation of the pathway, where we no longer have this pulsatile reactivation. And this really for us indicates that these phospho-ERK dynamics that we see are not driven by these transcriptional feedbacks, but rather by endocytosis and degradation of the receptors. And the last thing I want to show you is then predictions for drug combinations. And here I'm looking at combinations of the RAF inhibitor on the y-axis, the MEK inhibitor on the x-axis. And I'm first showing you this reference where we don't stimulate the system with EGF. And we look at 3 different conditions, and you always see the model predictions on top and the experimental validation on the bottom. And you see overall that there's pretty good agreement between model predictions and experimental data, and that in all cases, you see a shift in the potency of the response, both for cobimetinib, the MEK inhibitor, and the RAF inhibitor, vemurafenib. And the 3 conditions that we look at is on the one hand, the A375 cell line with upregulated EGFR levels on the left. Then we have in the middle a melanoma cell line with inducible NRAS mutations, which is the main resistant mutations that is found in patients, as well as a colorectal HG29 cell line, which also has a BRAF V6E mutation, which is quite similar to what we believe we believe that it's quite similar to the CRISPR upregulated EGFR mutation. And overall, this indicates that we can not only not only describe non-genetic or adaptive resistance mechanisms, but also these genetic acquired resistance mechanisms. And I think what this, on the other hand, also shows, and this is, I want to illustrate this a little bit with this allegory of the cave painting here, is that this really now gets to the summary. I think my summary slide somehow went missing while exporting this as a PDF, but what this effectively shows is that I think especially this last point about the endocytosis really indicates a little bit that the signaling that we see is a little bit like these shadows on the wall, and there's really some higher order organization in the system. And here we see that this is primarily endocytosis, which drives the signaling timescale, and there's probably other processes that also regulate this, meaning that it's not really kinase phosphorylation that determines the timescales, but really more higher order processes. And the idea now in my lab is to unravel these indirect observations through a combination of mathematical models with machine learning. And I think it's really important to have a multidisciplinary perspective on these things, and that dynamics are quite essential. This is why my lab is called Dynamics of Living Systems Lab. There's a master's student right now, Pierre, there's Marta and Giacomo, who are going to join me as a PhD and a postdoc. The stuff that I presented you today sounds interesting. Please reach out to me. I'm currently hiring both PhD and postdocs. And with this, I want to thank you for your attention. I'm happy to take questions.
Thank you very much, Fabian. Questions for Fabian? Yeah.
So thanks for the great talk. I have a question because of this BRAF mutation, and I think it's well known in the literature. And my question is, if there's this treatment, and does this give a growth advantage for the very bad cells when you treat the cells in the first case? So because the cancer is reoccurring and then the patients die very fast, would they also die that fast if you don't apply the treatment, and maybe these cells do not get growth favoring?
So if you have a BRAF V6 mutation, then you will end up with very high phospho-ERK levels, and if there's nothing else that prevents hyperproliferation, then these cells will die, yes. So you have to hit that mutation.
So it will be that fast in any case?
Yeah.
Thanks.
Thank you. Any other question for? Maybe I just have a quick one. In your network, you show many different loops, right? Feedback loops.
Yeah.
Is there any specific feedback loop that has a relevance for the adaptive state of the disease rather than others, or rather the control is distributed across different of these feedback loops?
Yeah, this is a tricky question. I would say that the inhibition at the receptor level is pretty important, and we here implement this through mainly through Sprouty, which is one of the receptor antagonists. But I think in terms of biology, the overall system is probably more complicated, but we don't really have the resolution to resolve it. And I would expect that it's a combination of multiple feedbacks that really is important for entering this drug-adapted state.
Thank you very much.